Good morning. Thank you very much for inviting me to this meeting. So it's really a pleasure to be here. My presentation today will be about screening for gambiense sleeping sickness. I will try to summarize what is available today, but also try to think about what is maybe still missing in terms of screening tools, in particular as we are now moving, as you know, towards elimination. So I guess many of you are already familiar with FIND, but I'll spend a few seconds just to highlight a few points about our organization. So FIND is an international organization, a not-for-profit organization based in Geneva. We were established in 2003. And our business model is really to work in partnerships. So we work with partners to try and facilitate or catalyze the development and also the implementation of new diagnostic solutions, specifically to address challenges related to diseases associated with poverty, including, of course, NTDs, but not only NTDs. We're also working on TB, malaria, Hep C, or HIV, for example. So I'll now move more specifically to the topic of this talk. On this slide, I've tried to summarize the key steps in the process of diagnosis of HAT. Diagnosis of this disease is still today quite complex. It involves three steps. First, screening to identify suspects. Symptoms are not very specific, so they have a limited value for screening. Screening is essentially today based on a screening test called CAT, which is an antibody detection test. Then it is necessary to confirm infection by microscopy to really demonstrate the presence of parasites. And finally, before initiating treatment, it's necessary to determine whether the patient is in the first or second stage of the disease, since treatments for these two stages are different today. So, as I mentioned, the test that is really routinely used today in Africa, and this has been so for several decades now, is the CAT test, the card agglutination test for trypanosomiasis. So this test has really played a central role in disease control and is still playing today a very important role. It has a number of important advantages in that it is cheap, it is rapid, it has a relatively good sensitivity and specificity. However, it also has a number of challenges, which are essentially operational challenges, such as the need for equipment, electricity, accessories, the need for a cold chain, and therefore it's not really an optimal test when we are thinking about the most peripheral and remote settings where this disease is found. And in addition, there is also a challenge regarding its production, since it's necessary to produce a native antigen which is obtained through infections of rodents. So, considering all these challenges, FIND started a project about 10 years ago to try and develop a rapid diagnostic test for HAT. So we did that by trying to involve and consider as many potential antigens as possible. So we screened the literature and were able to obtain 35 different antigens from 13 research institutes and universities. These were transferred to a partner in Germany to assess their reactivity using a panel of serum samples from cases and controls, which we obtained from the WHO and other research institutes. And based on this work, it was possible to identify 7 candidate antigens, which were then transferred for product development to South Korea at Standard Diagnostics. So, to make a long story short, this is what we call the first generation RDT, which was already developed and launched in 2012. It is today based on native antigens, so it's still a challenge, still a limitation. However, it has already met a number of significant targets in that it is cheap, 50 cents, rapid, 15 minutes. Important, of course, is that its sensitivity and specificity are roughly identical to CAT, and this is based on a multicentric trial that was conducted in 3 countries. And of course, since it is an RDT, there is no need for equipment, no need for electricity, no need for cold chain, and therefore it is already a major step forward in terms of bringing diagnosis more to the periphery, and also a good opportunity to try and integrate diagnosis and screening of HAT in the general health system. There is actually today on the market another test which is very similar to this test, which is produced by a company in Belgium called Coris Bioconcept. So this test is actually designed in the same format, using the same 2 antigens, and it has roughly the same diagnostic performance as was demonstrated in a study on stored samples. So we didn't want to stop there, and our goal was to develop what we call a second-generation RDT with 2 objectives in mind. So first, to replace the native antigens by recombinant antigens so that we would hopefully have a cheaper and easier to manufacture test. And also, the second objective was to be at least as sensitive and at least as specific as the first-generation test. So briefly, what we did to develop this second-generation test was first to select and clone new antigens, and that was done in collaboration with the University of Cambridge and the University of Dundee. Then produce recombinant antigens in South Korea at Standard Diagnostics, develop a prototype, which was then tested on stored samples at the University of Aberdeen and in Dundee. And finally, we conducted a prospective trial in the field to assess the performance of these tests, and this study was conducted in the Democratic Republic of Congo. So in the last minutes I have, I will try to summarize this study. So briefly, what we did was to compare the performance of this new test with the old one, the first-generation test, and CAT, with 2 objectives. So this was a non-inferiority study, so we tried to demonstrate that RDT2 was not less sensitive and not less specific than RDT1. For this, we included 15 sites, so 10 fixed centers for passive screening and 5 mobile teams in the province of Bandundu in DRC. We used as a reference standard a composite reference standard made of several routine parasitology methods. And we were able to enroll 260 cases and almost 57,000 controls. So these were the results in terms of sensitivity. If you look maybe at the blue bars showing all participants combined, first, the good news was that the second-generation RDT was apparently more sensitive than RDT1, so that was a key objective. But we were actually very surprised by the low sensitivity of all 3 screening tests. In particular, the CAT test, according to the literature, has been reported to be somewhere between 87 and 98% sensitive, and here we found only 62.5% sensitivity, so really significantly lower sensitivity. The same for RDT1, which in a previous study was much higher. So more specifically, looking at the differences, since this was the goal of our study and looking at non-inferiority, we were able to demonstrate that RDT2 was not inferior to RDT1. Regarding specificity now, results were quite good, with RDT2 around 98% compared to RDT1 at close to 99%, and CAT roughly in the same range. Again, looking more specifically at the differences between this test and comparing RDT2 and RDT1, we found that we were within our non-inferiority margin, which we defined as 1%. This both for all patients considered together, for active screening, but in passive screening, we found that the second generation RDT was roughly 3% lower than the first generation RDT. So now trying to think a bit more about the surprising results we got about sensitivity. What we did here was to consider the 260 confirmed cases and try to see more specifically by which test they were picked. And as you can see, while a number of them were detected by all three screening tests, many of these cases were also either detected by one or by two screening tests. So clearly, non-overlapping patients in terms of their serological profile. Which means that, of course, if several tests are combined, it gives a much higher sensitivity. For example, here you can see that if RDT1 and RDT2 are combined, then it's possible to achieve a sensitivity in the range of 90%, which is much higher than, for example, RDT1 alone, which is 59%. So going forward, that's maybe something that would be worth considering. Now, if we try to look at the underlying biology and try to understand why, more specifically, these three screening tests are giving so different results, I think it's interesting to look at the VSG code. So many of you may know that African trypanosomes have evolved a fascinating and very successful, actually, strategy to evade the host immune defense. And this is that they express a very dense coat of protein on their surface, which they are able to change during the course of an infection. And at any one time, actually, trypanosome express only one VSG protein, but they keep changing it during infection to evade the host immune system. And they actually have a huge repertoire of more than 1,000 genes encoding different VSGs, which helps them keep this chronic infection. So actually, although it's still a bit a hypothesis at this stage, but I think it's reasonable to assume that if we collect blood from a patient at different time points, we would get very different serological profiles because of different trypanosomes expressing different VSGs. And therefore, if we use only one RDT based on only one antigen, of course, depending on the time of infection, we may not get a very high sensitivity, which would explain why we would need either to combine several tests or, even better, if possible, to combine several antigens on one single screening test. Another possible explanation for this phenomenon might also be that since we have been using the CAT test now extensively in Africa for more than 3 decades, this could have actually caused a significant selection pressure on trypanosomes. And it's possible that during all this time, by detecting patients with trypanosomes expressing CAT antigens, we may have removed them a bit from the population. And today, the frequency of trypanosomes expressing CAT antigens, or at least expressing them early during the course of an infection, might be a bit lower than a few decades ago. And this, of course, would explain that today we have a lower sensitivity of CAT, and therefore, again, we may need to really think about new tests with new antigens to try to have a higher sensitivity. So to summarize some key points, so we have seen that we have today several options for screening. We have also seen that we were able to meet our targets for this study, which were to demonstrate the non-inferiority of the second generation RDT2. So based on these results, we decided that we would now register this new test, and it will be available and commercialized by Q3 of this year. We also saw that the sensitivity of all these tests today is still quite low, and this is probably because tests are detecting cases with very different serological profiles. And therefore, we believe that we should really consider the possibility of developing a third-generation RDT, which would be based on more antigens. Or, alternatively, we could also, at least in the short term, consider using several screening tests in parallel. Of course, that would be logistically a bit more heavy and also more costly, so it would be useful, for example, to conduct some cost-effectiveness analysis to determine also the best diagnostic algorithm, the best combination of tests, which is also likely to depend on the actual setting and in particular on the prevalence of the disease, and also if we are talking about active screening or passive screening. So finally, I'd like to acknowledge the very important contribution of many partners. So first, to provide clinical samples, antigens, to screen antigens, to develop the RDT in South Korea, to conduct laboratory studies, and of course also clinical trials. And finally, to thank our generous donors. Thank you.